Great. So glad to have you all here. Just in case you don't know this, we're talking about RIT and U of R. This is in Rochester, New York. And so I'm coming to you from my bedroom in Rochester, New York, the work at home life. So what I wanted to do here was give a pretty like basic 101, if you will, overview of what goes into creating an EEG-based BCI. This is in some ways a distillation of a lecture series I gave about a year ago at this point. That was over 3 hours long, and that went into more depth. But I'm going to try to fly through what you might need if you're just starting off into the realm of BCIs and specifically EEG. And an acknowledgement here: a lot of this information, a lot of the charts and diagrams you're going to see here are from this book, Brain Computer Interfacing: An Introduction by Rajesh Rao. And this is a great book I would recommend to anyone starting off undergrad level or early graduate level. But this book kind of goes through all of the subject areas that are involved in creating BCIs. So it's a great place to start. And a little bit about myself. I've been the project lead for the brain-controlled wheelchair team at NXT for about 2 years now. And I've been working more heavily over the past semester on getting a working BCI. And so at the end of this presentation, I'm going to give a pre-recorded demo of what we have there. That's my email. You can take that down if you want. I just wanted to say, if I'm certainly not an expert by any means. So if anyone here has a lot more knowledge in any of these areas and feel that I've made a significant error that's going to mislead people who are just getting into this, I would really appreciate anybody reaching out to me, and I'll try to issue some corrections afterwards to everyone who participated in this. Okay, great. So I'm going to take off into some basic neuroscience here, just the fundamentals of what you need to understand the rest of this lecture. So starting at a low level of what is a neuron, I liked Rao's, this is a quote directly from the book there, a leaky bag of charged liquid. So you have a potential difference due to a concentration of ions on either side of this lipid bilayer membrane. And we have these channels that control the flow of ions. And so the way that these neurons communicate with each other is what we call action potentials, or spikes, where you have a rapid influx of sodium ions. And so if you were able to record from an individual neuron from an electrode sitting right near it, this is what you might see. And these are what, again, we call spikes. And so there's some information there at a very low level about what is going on with individual neurons. And this is just an overview of what a neuron looks like. So we have the soma, the cell body, and it communicates, sends its spike over the axon, and it receives communications effectively from other neurons through dendrites. And now at a higher level, for what we're interested in with EEG, we're talking about the outer layers of the brain, so the neocortex, which is roughly an arrangement of 6 layers and 30 billion neurons, tons of connections between those neurons. And you actually have this pretty interesting columnar structure perpendicular to the brain surface. And what's important to know about the neocortex is you have functional specialization. So different areas of the neocortex are responsible for different things. And for the BCI that I've been working with, we're focused on the primary motor cortex, which is a strip roughly in this area here. And the summary, what you need to take away from this very basic introduction to what's going on in the brain, is that you have the communication with spikes within the brain, and it's the fundamentally electric nature of the communication between neurons that opens the door for BCIs. And specifically for EEG and EEG-based BCIs, it's important to understand the neocortex, so the outermost layer of the brain, and the functional specialization of the different regions. So now, how do we record? How do we pick up information from the brain? And this is where electroencephalography EEG comes in. So for EEG, we have electrodes that are sitting on the outside of the scalp. So this is a non-invasive technology, no surgery, nothing required. And actually, this is a good time as any to show it off here. This is the OpenBCI Ultra Cortex, which is the structure we've been using for creating BCI. So it sits on the head like this. So very simple to take on and off. It isn't the most comfortable thing in the world, but it gets the job done. And the electrodes, if you can see here, these are spike electrodes that will actually try to go through your hair to make a good contact with your skin. And there's flat electrodes as well to go against forehead or any exposed skin. And these are just spring-loaded electrodes. They're actually just plastic, but coated in a highly conductive. It has a highly conductive coating. So for EEG, what we're picking up information from large populations of neurons. We're definitely not able to see what we saw before with those spikes, the communication between individual neurons. And we're not able to pick up information from too deep within the brain. Like I said, we're looking at the outer layers. And this is really because we're talking about voltage fields here for what we're picking up. And that's going to fall off with the square of distance. So you can't get too far down. And when we're talking about EEG, usually we say that it has a good temporal resolution, where activity within the brain is able to be picked up very quickly. But you have poor spatial resolution. So really you're talking on the order of a square centimeter for what's a useful range to pick up data from. There is effectively a maximum number of electrodes that you can have before each one is just picking up such similar information that it's not going to help you out. And what you have over to the right here, this is what's called the 10-20 system. So it's a standard and agreed upon set of electrode locations. Some of the important ones that we're going to be looking at further here is C3 and C4. So these are over the primary motor cortex strip that I mentioned earlier. And so from C3, we're actually going to be able to see some information about what's going on with the right hand specifically, and C4 for the left hand. So that's what's going to go into the BCI that I'm going to show you a bit later on. We're talking really low amplitude signals. So these are low voltages. They require strong amplification. In our case, we're using the OpenBCI site on board. I'll have a picture of that coming up here. Since the information that we're picking up from the populations of neurons is not strong, it's easily contaminated by muscle activity, electrical devices, some things that you might consider user noise. So if we say noise is anything that we're not interested in seeing relative to what we would say is our signal, some forms of user noise are eye movement, eye blinks, facial muscles, any muscles on the head, and head movement. That's one of the things that we get with this OpenBCI Ultra Cortex is there's definitely a bit more shifting going on if you're moving around compared to compared to something like this cap that you see on the right that's going to be more secure under movement. And psychological states actually will change what you are seeing in your EEG data. And so depending on what you're doing, you might consider this noise, or in some cases, you might actually be able to try to determine what these states are. And for non-user noise, power line noise, very common. I mean, this is going to be the strongest thing that you see in your data. And so it's very common to just throw a notch filter in to filter out at 60 hertz or 50 hertz, depending on where you're living. And electrode impedance can also change over time as you're wearing the headset. And so that will change what we're picking up from these electrodes over time. Sometimes people abrade the scalp to get a better connection. I found this wasn't necessary with what I'm working with here with the OpenBCI electrodes. Again, I talked about this 1020 system, and also I mentioned that you do have some high density EEG arrays, and you're going to see a lot of the same information coming from two electrodes sitting next to each other, but there's a lot of awesome techniques to determine what is unique to each and how that can inform your system. So now here's a picture of the board. It's actually sitting on the back of this this ultracortex here is the Cyton. It's in an enclosure. But the picture on the right is a Cyton board with what's called a DAISY module attached. And so the Cyton board by itself, you're going to be able to record from 8 electrode locations. And when you use the DAISY board, you get up to 16. And there's a few different ways we reference our electrodes. One of the most common, and if you're looking through the OpenBCI guides, this is what you're going to see, is these bottom pins actually, or I think you can use either of the pins if you're only using one, but they're going to be referenced to ear clips. So you have 2 ear clips in the case of the OpenBCI system. You see him hanging down there. One serves as the ground for the system, and the other serves as a reference point for all of the other electrodes. So this is a differential amplifier system. So it's amplifying the difference between your reference and the electrode of interest. So that's what we would call a unipolar electrode configuration, or you might also have a bipolar electrode setup. So if I flip back to the slide here where you can see the 1020 system, you might have a bipolar setup where you're actually recording the difference between, say, C3 and CZ. That can give you some different information. And so the OpenBCI Cyton is actually capable of this by plugging into both pins here. So if you have leads connected to both of these pins, it'll do a difference between those rather than a reference on the ear. And there are some other referencing methods as well that are typically done in software, not at the hardware level. One of the awesome things that we get with the OpenBCI Cyton and probably a lot of other boards is all of the electrodes are sampled simultaneously, which means that theoretically we can go in and rather than have 2 leads connected that it's taking the difference between, like I said, C3 and CZ, you can actually just subtract those. You can take the difference of them in software and you should get the same result. So you can do more advanced referencing. There's common average referencing where you're referencing against the average of all of the electrodes or just some of them. And then there's another type on here that's not on here called a Laplacian, where it's looking at the nearest neighboring electrodes to do that reference. So again, we're talking about pretty weak signals. So your amplification might be 1 to 100,000 times. And typically the sampling rates you're going to see are from 250 to a kilohertz. And the OpenBCI Cyton board is capable of 250 hertz in its standard configuration. If you add that DAISY module, you drop it to 125, so you half it. This is a limitation of the Bluetooth, the low energy Bluetooth dongle. So this is a wireless board, and that's the USB dongle that gets plugged into your computer. They do have a Wi-Fi module as well, if you wanted to use that instead, and then you can get up to the max that the chip is actually able to handle, which I believe is 1 kilohertz. And what EEG data is really well suited for observing is oscillatory brain activity, which we call brain waves. And we can get some information from the synchronization or desynchronization of populations of neurons. And so when we're talking brain waves, we have these classifications for different frequency ranges. And so a while ago, when EEG was more in its infancy, there were plenty of doctors, primarily medical doctors, who were observing this EEG data in its time domain form, like what you're seeing here on the right, and trying to determine what's going on in that person's brain over different regions. And so there are actually books just filled with different waveforms, effectively, from EEG data that try to determine what might be going on. So, anyways, these are the different classifications of brain waves to different frequency ranges. I would warn that with these alpha, beta, delta, they vary from study to study sometimes. There's actually a paper, a review paper that goes through and looks at how these vary. So they're not terribly consistent. So when in doubt, I would look more at the actual frequency rather than those names. But our different frequency ranges tell us different things about what's going on in the brain at any given time. So some things like drowsy driving, you might see more activity in the lower frequency ranges, the theta range. And so just a summary of this, what we need to know going forward is we're recording electrical activity of populations of millions and billions of neurons. And these populations of neurons synchronize under certain conditions, allowing us to detect these brain waves. And we're really trying to characterize the brain waves that are detectable over certain regions. And researchers have been doing this for decades now. All right, so now for the main event, jumping into EEG BCIs and how you create it and what kinds of EEG information we're looking for. So this is, again, from the Rao book, a representation of the components of a brain-computer interface system. So we have our signal acquisition phase. So this is our EEG differential amplifier board picking up data. And then we go through a process of pre-processing and feature extraction. So you might do some filtering. You might compute some features in the time domain. You could do Fourier analysis and take out some features in the frequency domain. And a lot of the techniques that you'll see done, certainly in the last couple of decades, have really focused on different machine learning techniques, where you're talking either classification or regression. And Yeah, so you can classify those features into potentially two or more classes that allow the system to understand what's going on. And so really what's enabling this is our brains are good at learning an action to receive a reward. There were some very early experiments with primates actually recording from individual neurons. So this is invasive, looking at the spikes of an individual neuron and determining that that those primates can actually learn to modulate the rate of firing of that neuron to receive a reward, which is very cool. And from an EEG perspective, what we're talking about is learning to modulate the activity of neuron populations. So two broad categories of approaches for EEG BCIs, or all BCIs really, are a self-paced or asynchronous mode, where the subject, the user, is voluntarily modulating those neuron populations to do something like move a cursor or select one of the options that's on a screen. And then there are stimulus-based or synchronous, a synchronous mode, which is the BCI system detecting some brain response that is generated by another external stimulus. And typically those stimulus, you'll connect it to a certain choice. So it might be a word, or sorry, a letter and a speller, or a certain option on a screen. And so the point to take away is that this isn't, this synchronous mode is not initiated by the subject.
Hey, Adam, if I can interrupt you quickly, we have a question in the chat. I think you might answer this later, but okay. Someone says, which software do you use, EEG Lab or MNE Python, ?
Cool. Yeah, so I use, yeah, I don't know if I've tried EEG Lab, but I do use MNE Python for analysis after having collected some data. I think it's a great, it's really a great open source tool. It's definitely something you should know about if you're interested in this area. What I use for my real-time, if you will, BCI system is a package called BrainFlow, and this allows you to pick up information from various. It's board agnostic, so you can pick up EEG data from all different kinds of boards. In my case, the OpenBCI Cyton board, and it also has some signal processing tools built in. So that's what I use for real time. Cool. So oscillatory potentials and event-related desynchronization. So in this context, we're talking about imagery or motor imagery-based BCIs. And in the case of event-related desynchronization, which is actually what's enabling the BCI that I'm going to show off here in a little while, is when you imagine movement of your right hand or your left hand, that is going to be detectable as a desynchronization. So really a decrease in amplitude for a certain frequency range over the C3 and C4 electrodes that I showed before in the 1020 diagram. And so some of the earliest experiments with this is cursor control. And this is focused on the mu band, which is 8 to 13 hertz. So somewhere around there is where you're going to see your most important information when you're talking about motor imagery, event-related desynchronization. So really, when you're doing this, you're thinking, you're imagining the movement, but you're not performing it. So here's one of the earliest cursor control experiments. This was actually performed in Albany, New York, so very close here to those of us at RIT. And this was 1D cursor control with just a few subjects, or 4 subjects. And it was using 2 electrodes, anterior and posterior to C3. So it was a bipolar configuration like we talked about before, where you're taking the difference between those 2 electrodes. And it was looking at the amplitude at the 9 hertz frequency band, taken in 1/3 of a second windows. And the amplitude of that 9 hertz band was mapped to different movements up or down of the cursor. And so in this case, I believe it was when you're relaxing, or that's what they trained the users to do, is relax. And that actually really raises the amplitude, the power in that band to its maximum. And I think in their case, it was moving the cursor upwards towards the target. And then the event-related desynchronization. So the user imagined moving their right hand. Some people imagined lifting weights. That was a downward movement. And we saw a decrease in amplitude in that 9 hertz band. So this chart really shows this well, but it might be a little confusing to look at. So I'll try to explain it here. So the x-axis here, you're looking at the amplitude. Again, this is just 9 hertz. So the amplitude at 9 hertz, so low to high. And the y-axis is the percent of all of those amplitude recordings for each 1/3 of a second time window. So really, this is a density plot of all of those recordings throughout the trials. So notice here that the black line, this is the event-related desynchronization. So the motor imagery of moving the right hand resulted in lower amplitudes at that 9 hertz band, which is exactly what we want. And the dashed line is the relaxed state. And so what's important to see here, you can see it shows a correct rate and a number of hits per minute. And so you can kind of see that the more distinct these 2 classes are, either relaxed or in the motor imagery state, the better performance that user has. And this is the step ranges. So the way they set it up is you kind of have this dead zone in the middle where it might be your motor imagery, it might be you're in the relaxed state, so the cursor isn't moving. Or if when you get down further, lower amplitudes, then you're moving down to the target. And then they have 3 different step ranges to move up towards the target. So really, you're only seeing these higher amplitude values in that relaxed state, so you can make a bigger movement towards that top target. Looking at the chat here, yes, this recording will be available to watch later on. We're going to have it posted on YouTube a little while later. So, yep, okay, I think that's it for that. And this is really what the BCI that I'm going to show is trying to replicate. But in my case, I'm using, I mean, it's 30 years later, but I'm using significantly cheaper hardware and something that you can buy off the shelf and have very quickly. And it's a completely dry system, so you're not using electrode paste and getting gunk in your hair, if you will. So that was just a one-dimensional cursor control, but they upped the ante to 2D. This was many years later, and it involved more electrodes, and they used, like I said before, that Laplacian filtering technique around the C3 and C4 locations. So notice that we have C3 and C4, so right and left hand motor imagery. So this enabled 2D cursor control. So that's what this stepped into. And rather than having those kind of bins to determine how far you're going to move the cursor, they actually had continuous equations that had weights applied to the value of the C3 or C4 amplitudes. And they got, again, just 4 subjects, but they got them all to 70 to 92% accuracy, which is certainly impressive. You can see what they did here with 8 different target locations. And so this is those 4 users. User A and user D, you can see had really great control of that cursor, went pretty much directly to the targets, and somewhere around a second for each of them too. So that's awesome. The other ones did acceptably well, I would say. There's some squirrely pathways like this one here, where you might get in trouble in a real world use case of this, where you go over a target that you didn't really want to. But really impressive results with still not a very complex system behind it. So this is a different type of potential. So the event-related desynchronization from motor imagery, you're able to detect that for an extended period of time. That desynchronization, that decrease in the amplitude and that mu frequency band. But there's this other potential, movement-related potential, which is also called the readiness potential, and it is a more significant but ephemeral, just a short deflection in the primary motor cortex. So it's detectable as the preparation of a movement, but the execution of that movement doesn't actually need to occur. Sorry. Now this is moving into the synchronous modality that I said before. Sorry, looking at the chat again here. So I certainly haven't gotten into 3D at all. I'm not sure of the latest research looking into that.
Not so much that I've seen with motor imagery, but definitely some SSVEP applications. So it's getting back to the synchronous BCIs, just because you can add more elements to look at. And there are plenty of invasive systems that have been doing that for quite a while. Miguel Nicolelis comes to mind. I can put a link into the chat. So there's like virtual control, and then as well as robotic prosthetic control. So I'll post a couple links in the chat.
Cool. Thanks, Harrison. Yeah, so this is synchronous. So it's detecting some response of the brain as a result of some stimulus happening on a screen or somewhere else. So the stimulus evoked potential, there are potentially many different stimuli that work for this, but the notation that you'll see is P followed by a number of milliseconds, or N, and that's either a positive or a negative deflection in that EEG recording. And one of the most common forms of stimulus evoked potential is the P300 speller. So again, this is a positive deflection 300 milliseconds after the stimulus, roughly. So you can see these deflections here, just an example of it. So the P300 speller, I'll pull up this video and talk over it. Is it has all of your letters, and then your flashing rows or columns, and the user, the subject, is going to focus their attention on the letter that they want to be spelled out. And after enough flashes go by, the BCI system is able to detect which letter they had their attention focused on. And moving on to what Harrison just mentioned. So this is a steady state visually evoked potential, SSVEP. So rather than just a quick flash of something that they're focused on, it's a constantly fluctuating target area on the screen, usually at a rate greater than 5 hertz is what works well. And so for this, we're looking at EEG electrode recordings over the occipital region of the brain, which is in the back. And you're actually able to detect that same frequency that you see on the screen from those electrode locations. So this was first explored in the early 2000s, and you can see a typical format for this will be a bunch of buttons or menu options on a screen that has some element flashing at a certain rate. Another one. So rather than a visual stimulus, it's an auditory stimulus. And so rather than focusing your attention something on a screen, you're actually listening. You're attending to either the right or left ear, and short beeps in either. And so you are able to detect a similar deflection that you would see from the stimulus evoked potential, like that P300 speller. Air potential is really interesting and has really been studied more just over the past decade or so, so more recent. But it's the brain has detectable response to misclassification, whether it's in the use of a BCI or something else. So one of these early studies was actually just controlling a robot with a controller. It wasn't a BCI system controlling the robot, but it was just a controller. And every so often, that robot would make an incorrect move. And sure enough, with the right electrode locations, in this case CZ and FC, looking, I guess it was somewhere in the 1 to 10 hertz range, you're able to see this pretty distinct waveform in the time domain. And one of the things that I saw from later research was that this error potential, as it's called, can actually be classifiably distinct based on the type of error that's made. So potentially, in the event of using a BCI, there are different types of errors that happen in the world, and you're able to detect each of those uniquely as error potentials. And so really you have the opportunity to try to apply this to your BCI system to detect when something went wrong and figure out how to remedy that. And there's a topic of co-adaptive BCIs, which is really so on the BCI side of things, within the system, within software, we're looking at machine learning. So so algorithms that learn over time to become better at classifying the user's intent. But we're also dealing with human subjects who will change their strategies over time and get better at modulating the activity of neuron populations. So co-adaptive BCIs, it's a discussion about how do we optimally let the user change their strategy and improve themselves while also improving the BCI, the software side of the system. Just some reasons why the EEG data might change over time. So again, user like change in strategy, fatigue can change the EEG data that you're getting, changes in electrode impedance or location. This is one of the problems that I have with this OpenBCI system is, well, it's just me, and I don't have anyone sitting around able to measure to ensure that this C3 electrode that I'm interested in is at the correct spot. I tried to get the headset on consistently, but sometimes sometimes it doesn't go on the way it needs to, and it becomes difficult or impossible to exert the control that I have in other circumstances. And yeah, so this is just a study that really explored how you can do this effectively, and it showed that it doesn't require tens of hours of user training to become effective at controlling a BCI. But if you have good learning algorithms on the other side, you can, within just a few minutes or really up to an hour, for people that are struggling more, exert a good control over your system. Okay, so that's the end of kind of the foundation of what I wanted to cover. Hopefully that was informative introductory level. And now I just wanted to show what I've been working on really just this past semester for an actual BCI implementation. And what I wanted to do was effectively replicate that one-dimensional cursor control that we saw with the Woolpaw team about 30 years ago now. But again, we're working with hardware that is probably a fraction of the cost that theirs was at that time. But thanks to technology improvement and the work of OpenBCI, among others, we get this pretty awesome hardware at a much lower cost. You're talking somewhere on the order of $1,000 for this system. So really wanted to just validate that with this lower cost system that we can get pretty much right off the shelf, that we have multiple of within the NXT organization, that we're going to be able to see this data, this EEG information that other researchers have been. And so there are some modifications to what they were doing in that system. I'm using the power spectral density rather than just looking at the amplitude for a specific frequency band. In their case, it was just they were looking at 9 hertz. I'm looking at the PSD, power spectral density, for 10 to 12 hertz. And roughly every 1/10 of a second, I'm recalculating the PSD value over the last 3 seconds of EEG data. And there's some windowing going on in there, but that's a bit technical. And like in the cursor control study we saw before, where you had the bins determining how far the cursor is going to move. In this case, I had a similar binning solution rather than a continuous equation, and it changed the velocity of the cursor to move either up or down towards the target. So on the next screen, I'm going to show a video demo of what we have. But I wanted to show what the user interface looks like before we get into that, because it is maybe a bit to take in. So just at the top, there's a running tally of how many targets we've hit and how many we've failed on. Each trial, the cursor will start in the middle, and I will have 10 seconds to get to the target. There's no penalty. Important thing to note, there's no penalty for going where the other target would have been. So this target either comes at the top or the bottom. But actually, you'll see that actually in this video, I don't think there's really any case where I would have hit the other target and then it comes back down. But most of the time, if you do, you're not able to control it well enough and you go up into that region, you'll run out of time before you can get back down to it. On the right, you'll see, again, the one to focus on is the 10 to 12 hertz. That's what's informing the cursor velocity. But these are your other common brainwave ranges. And this is a frequency domain plot of the power spectral density, with the green highlighting the 10 to 12 hertz that we're looking at. So if there aren't any questions about what you expect to see there, I'm going to go ahead and play the video. What you're going to see is 20 trials. And well, I'll let you see the result. So what you're going to see if you look over towards this power spectral density graph is when I am trying to do the motor imagery is when the cursor is going up, and so you're going to see a lower value for PSD in that range. To move the cursor down, I'm effectively relaxing and trying to maximize what we're getting in that frequency band. And this actually is one of the bugs that I had in the system. This only happens once, fortunately, where the cursor actually it only updates a few times per second, and it skipped right over the target. But this is a log scale over here, but hopefully you can see that we are getting the higher levels when the cursor is moving downwards and I'm relaxing. This is the last one here. So there you go. Yeah. So hopefully you could see that okay coming across on Zoom. But that was 18 successful target hits out of 20. And oops. Sorry. There we go. And so I wanted to compare my result with what I showed you earlier, what I explained to you earlier, this graph of the 1D cursor control from Wolpaw. So again, this is showing the density of the recordings that we're getting, the power spectral density values that we're getting when the target's at the top, which is the blue, and that's me doing motor imagery. And so you have the event-related desynchronization and the lower PSD values. Whereas when it's in the bottom and I'm relaxing, you get a wide range of values, but it's flatter, and you get more towards this high tail end. And so that's when you have your big moves downwards with those different bins. So it was just awesome to validate effectively the hardware that we had against this study that was done 30 years ago at this point. One of the real challenges was, again, sometimes I would put the headset on and it just everything would just be in the right place and recording well, and this would work effectively. And other times I wouldn't be able to exert much control. So that's one of the challenges of the hardware that I experienced, but that's the result from that. So now I'll take some questions. So these are, the question is, are your headsets commercially available? So these are OpenBCI. All of this hardware is from OpenBCI, which is actually, as the name suggests, it's all open sourced. But they have a store. It's a company. They have a store. You can buy all of this hardware off the shelf. Yep, Harrison just linked it there. I'll show it again.
That was their donation link. Apparently that's what comes up first when you click on it. I'll get the other one in a second.
Okay. Yeah, so there's a link there, but this is the frame here, and this is actually 3D printed. So NXT has a 3D printer. We 3D printed the frame to save some money instead of buying it from them. So this plastic frame, and then it has these screw-in electrode inserts. And so we purchased the board. We didn't make our own. We didn't purchase our own PCBs and populate them. We purchased the Cyton board ready to go. But we also purchased these electrode units, which have threads and they screw in to that frame. And these you could try to 3D print them and make them yourselves, but these are injection molded, and they're much stronger than what you'd be able to do with a 3D printer. So these we paid for it. They work well for us. And the frame comes in different sizes. What you saw there was a large, which in some dimensions is actually a bit big for me, but a medium is the right size for most people. Yeah, if anyone else has questions, you can throw them in the chat. You can raise your hand in the participants tab and then talk if you want.
While we're waiting for some questions to come in, I just want to say that was an awesome presentation. So hats off to you. And I hadn't seen your demo yet of that. So congratulations on that accuracy as well. That was very cool. Very cool to watch.
Yes, it was. It felt great to get it done. We've been talking about this stuff for a while and learning the fundamentals, but to really be able to pull off a system, although it's still a very simple system, to validate it, it's great. And it's a very neat experience to actually be controlling a BCI system. So. All right. If there aren't any questions, I guess I pretty much nailed the hour time. So thanks everyone for joining in. Again, we'll be making a video recording of this, and we'll get that sent out over some channels if you want to review anything from it. But thanks all for joining.
Yeah, so as Adam said, that will be going up on our YouTube channel. We'll also put links to